Okay, good morning. Wonderful to see so many faces in here. A fantastic meeting, amazing city to be in, and our thanks to the organizers for this fantastic event. Over the past 2 days and prior to this meeting, we had a fantastic interaction with the exchange team that is with us in here, a beginning of a discussion on big data in metabolomics, biorepositories. The use of this big data was a great, I wish we can give you a bit of a summary of this again. So we have a marathon to run very quick as we go through our program today. The center of this session is to leverage 20 years of metabolomics data that we have been generating, or however many years you have been in this field. We want to use the collective data that is out there towards knowledge. And in the case of human diseases as applications, we want to use it to apply it to understand human disease, disease progression, subtypes of disease, response to a treatment, and so on. So we want to be able to use the collective data, in our case, the brain. We have been studying CNS neuropsychiatric diseases. So we want to be able to use the collective data that is out there, what we have generated using different platforms, what is available out there, and collectively, as a community, learn together. So the session in here is going to talk about the quality of the data, what is uploaded to the biorepositories. We are going to hear a bit from Rick about a reactivation of the minimal, what we started 20 years ago, over 20, we're getting old, what we started with the beginning of the Metabolomics Society. We started in 2004, 2007. We had a minimal standards initiative. Rick will talk about this and the reactivation of this 20 years later to try to address issues related to standards and standardization of processes. So whatever data is out there, we can at least understand how it was generated, how to use it. Jennifer is going to address QA, QC, a fantastic effort by a working group that has been doing diligent work. Han is going to give us a perspective on the Lipidomics Society and the Lipidomics community and how they try to tackle issues of standards and standardization in reporting lipidomics data. Peter Meikle will illustrate in practical ways how do we connect data and harmonize data. If we are using 6 or 7 platforms that are giving us a read on the lipidome, how do we connect all of this data and use leverage the lipid part of it? Then we have 2 talks by Martin and Tuulia to add to the complexity of the metabolome, the exposome, which was touched upon again yesterday, and certainly a big space of biochemistry to cover. The FAIR data, a future perspective on FAIR data, data visitation, digital twinning, making data ready for AI is a futuristic, and we have a fantastic panel of discussants, Susan Sumner, Tuulia, Mate, and David, to try to have a discussion at the end, 20 minutes. So we're going to run 10 minutes per talk, 2 minutes questions, and from there, I don't know how to move forward. I'm going to move from this. I need to get into my talk. Just 2-3 slides. We covered the exposome. Just in thinking about the human disease, human diseases are complex. They are influenced by the genome, and this is what we have been studying for a long time, but the genome doesn't explain everything. The exposome, the diet, the lifestyle, the drugs, the supplements that we take, the socioeconomic influences on our health, all of this influences our metabolome, and therefore we see the metabolome as a readout for all of these net influences. And by using our tools and capabilities as a metabolomics community, we are trying then to define these changes in the metabolome and ask questions. If I see a change in a disease, is this related to the genome, the exposome, the gut microbiome? The gut microbiome is a big influencer, so we want to put this all in context as we think about the signatures that we are seeing in human disease. So the metabolome is powerful, it's dynamic, and it is giving us a read on our health point by point in real time. So between the molecular mapping using the metabolome and the digital recordings, we are truly starting to think about changes in health and in real time. All right. So we talked then about the genome, and then everything else currently we are putting under the exposome, whether it is the chemical exposures, the diet, the drugs, the gut microbiome, socioeconomic. So it's a big, big, big bundle of things we are calling the exposome for now. For now, and it is a lot of things we see in the blood as we profile human samples. Some of the questions we were raising as a community, and we would continue to debate throughout the session, we want data that is usable, reusable, with good quality. So issues, we talked about issues of quality. We want to keep in mind the data comes from a patient or from an individual. So truly, everything we are using and doing, is thanks to the people who are donating the samples, who need to know something about their health. So we are going to put the patients and the individuals who are providing this data central and trying to help them in every way, right, to learn from the data that we are using and analyzing. We have different types of platforms we use, whether it's NMR or mass spec based, and the collective power of all of these platforms is a great power, but adds complexity. For the people who come from the clinical field, they say, what the hell are you using? You have too many platforms. Which one should I use? And what does it tell me? And how do we connect if I'm measuring data from 8 different metabolomics platforms on the same individual? How do I even put this data together from one individual, leave alone putting data from different individuals? Yes, the FAIR data, we talked about it. I'm just quickly going through my sustainability. How do we keep this big data sustainable with the funding that comes and goes? We discussed this yesterday. We're going to continue to think about it. We need to have resources that can keep this metabolomics data for use and reuse with some good funding. Harmonization, we said, is a big thing. Globalization, a global perspective. Definitely want to keep this global perspective. Acknowledging the people who give and donate the data. If I'm using data someone else produced, we want to make sure we have rules and regulations that can acknowledge the original source of the data and the people who put blood, sweat, and tears in getting this data available. And importantly, we want to protect the identity of each individual. So to upload data to any biorepository, you need an army of people to make sure if it is a human data, it's de-identified, we are okay, we are in a safe zone, no one can connect it back to a subject or to an individual. With this, we are gonna move on now to our talks. We are gonna start with Rick and talk about the standards initiative, a look back and a look forward. Exactly 10 minutes, please, per speaker, otherwise we're gonna get you off stage. 2 minutes for questions, and then we wanna leave a time for the discussion with the discussants.
Thanks, Rima. Apologies, so if anybody was in workshop 2 yesterday, it's a very similar talk to that, but hopefully most of you won't, so you're going to learn something new. So we're going to talk about data reporting standards. So this is reporting about the methods, the subjects, how the data's been collected, how the data's been processed, etc. And it adds that interpretive layer onto just submitting data. So everybody can submit data, but if you can't understand that data, you can't check the quality of that data that Jennifer will talk about later on, then the data's not necessarily reusable in a way that you want to do that. So I'll talk about, actually I'll skip over this because I've only got 10 minutes. I thought I had 12 minutes, sorry. So data reporting standards, they want to provide a description, which hopefully is machine readable. Now that's a question to be asked and discussed later on, which talks about the experimental study, how the data's been collected, and the biological subjects within there, whether they're humans, plants, microbes, whatever within there. And really it's a standardized reporting, so we can compare and contrast across different studies within there as well. So data reporting standards should feed into FAIR principles, okay? They have to, and we'll hear about that later on from Thomas within there. And really, so we're thinking about standardized reporting frameworks. So if there's a standardized way that you can easily provide that information, and I know there is some, whenever you do this first time, there's always some pain within that, learning how to do that. We're looking at that at the moment, as you'll see later on, but also once you've done it once, it becomes a lot easier because a lot of it is copy and paste, etc. within there. So do it once, learn about it, becomes a lot easier later on. So what are data reporting standards not? They're not defining how you should collect your data, how you should collect your samples, etc. It's definitely not. A number of people have discussed this with me and we're definitely not requesting that. So it's not the standardization of methods that you will apply in the lab, for example, within there. It's the standardization of reporting of all those different methods that you're applying along with the data, along with the subjects, etc. within there. So we're not telling you what to do, okay? We're asking you to be able to define what you have done. Why do we need data reporting standards? Well, we're talking about, I'm here for the Metabolomics Society, my Metabolomics Society hat on, but actually, in what I'll talk about in a minute, we're looking across the whole small molecule regions. So we're thinking about metabolomics, lipidomics, and exposomics is in there, as well as some other small molecule analyses. So we're not just talking metabolomics. But we know that metabolomics, lipidomics, exposomics data are an important source of new knowledge. A lot of data is being deposited, for example, in the data repositories within there. And with the data standards, really, we can use that data. We can go and reuse that data within there. So if we understand how the sample's been collected, how the data's been collected, what each sample or each sample file is, data file is, etc. within there, whether it's within a single study or multiple studies, that really helps us to be able to understand and to reuse that data, okay, within there. One thing which I'm not going to talk about in depth, but if you go into a lot of the data repositories, go into the data, most of the data that has been deposited, it's the biological data. There's no data for QC samples, there's no data for blank samples, etc. But that's really important to include, to look at the quality of the data. I think Jennifer will talk about that. So there are different data reporting standards out there that have come across from the last sort of 17, 18 years. I'm just going to briefly introduce all of these. The first one was the MSI, the Metabolomics Standards Initiative, not mass spectral imaging, okay? So just make sure you got the right frame for that. So these were developed from the Metabolomics Society in 2000, started in 2005, were published in 2007. And these were all the papers. So there were 9 papers published in there. There's a lot of people involved within this, and credit to everybody that was involved, okay? But I've just put the citations in there. So this is 18 years ago. Every time you use reporting standards, you should be citing it. So we're not necessarily using reporting standards routinely in metabolomics at the moment anyway. And this really was focused on metabolomics. And you'll see as we go through the different data reporting standards, they have different concepts, different contexts within them. And then the next one, I'm going to try and do these in order, they're not fully in order. So the next one, again, was metabolomics, but it had an application context within it. So this was with regulatory toxicology. So this is from Mark Vine's group at the University of Birmingham and others as part of the Merit project. So this is really focused on reporting standards and best practices for regulatory toxicology metabolomics, so application specific. The third one, and I really do like this one when I use it, is the Lipidomics Standards Initiative. So this is an online, basically checklist that you go through and that you can download at the end of that, and then you could submit when you're submitting data into data repositories or into papers as well. So it's a checklist. It's composed of 9 sections. It is relatively easy to complete. It's relatively easy to understand, etc. as well. So it's the quickest I've done of any of the reporting standards to be able to complete that. mQACC, and I think Jennifer, wherever she is, will probably talk about these later on, so I'm not going to talk about these in detail, but mQACC have developed reporting standards to report on a specific part of that experimental workflow, the use of QA and QC within the study, mainly around pooled or quality control samples, but other aspects of it as well, and I'll let Jennifer talk more about that. And then BP4NTA, which is a group mainly based, or actually all based in the USA, I think. Apologies if I'm wrong on that. And they have a study reporting tool. So again, this is online. You can download it, you complete it, you can submit it as well within there. So all 5 of these, they have different context within it, or concepts within there. They will have different advantages and some limitations as well within there. We're not, within what I'm going to talk about now, really thinking about, we want to develop one set of reporting standards. It's not what we're thinking about within the group that we've got together to look at these different reporting standards. Each of these has different applications within there, and we want you to use the appropriate one, whichever one that is, okay, within there. But really, we're here to think as one voice, to communicate that you should be using reporting standards, but also think about, do we need to make any changes to these reporting standards within there? Now, I think there's one important thing that over the last 18 years has been very much a carrot. It's your choice whether you use those reporting standards or not, and whether you submit that with data to papers, to journals, etc., or to data repositories. And most people aren't using that, we'll be quite honest with that. So now it might become more to a stick where you will need to do that, and we see this in other omics. Okay, you have to submit specific information when you're submitting data either to journals or to data repositories. So I think that's probably the way that we need to go, but that's my view. We'll see how that goes. So we've got a number of different stakeholders in here. So we've, as you'll see in a minute, we've got a group of about 50 people across all of these stakeholders and elsewhere as well. So we've got the Metabolomics Society, we've got the International Lipidomics Society, the BP4NTA, mQACC, etc. as we go through all of these. The funders as well. So we have a representation, for example, from NIH on there. Within there. And so we're looking at this in a range of different ways. So we've developed a group, we've come up with a name, which we voted for literally in the last week. We're going to be called PRISM. So this is going to be precise reporting in small molecules. And then we're going to have the date after that. So it might be PRISM 25, 26, etc. if we have revisions, etc. as we go forward. As I said, we've got members across all of those communities. We've had one stakeholder meeting in January this year. We've broken up into a smaller subgroup where we're looking and comparing and contrasting all of the different reporting standards that are out there. And we're hoping that that will come together and show what the minimum reporting standards should be, i.e. what's being used in all of those different reporting standards that I've just spoken about within there. So we're now working on that. Hopefully that will be available later this year, and then we'll move forward from that. But we are here for one voice for data reporting standards across these different communities, and that's important. So I'm here talking today, but this is a big community, and thank you to everybody that is involved in this, because it is a really important topic. So we will keep you updated through the Metabolomics Society, through the other societies as well. But please, whenever you're submitting data, okay, whether it be to a data repository, whether it be to a journal, please think about using one of these reporting standards and submitting that with your data or with your publication, okay, within there. And exactly on 10 minutes, so brilliant.
One or two questions, maybe just brief, short, and if not, we can leave the questions for the discussion at the end. This is your society, it's your voice, it's a podium for all of you. Why should you share data? We're gonna discuss this as we go along. It makes the impact of the data that you generate enormous, far more than any one of us can do alone. We want to be able to come up with a reward mechanism where every time someone uses your data, you get rewarded. If you have analyzed the data and you did not analyze it in depth and someone adds to it, we want to make sure you get the credit. We want to come up with an index that gives the reward, so this becomes a fantastic process. Please give us ideas, shape this initiative and the initiatives you will hear about as you see best for you and for the community at large. Rick, thank you for leading this again. 2007 was a great effort. Did it have a big impact? We tried to go minimal standards, minimal hand-holding. It was useful, but I think we need to do more. All right, Jennifer, quality of the data, a perspective. Thank you.
Hi, everybody. I don't know whether it's possible for me to have slides. First, failure, not understanding the technology. You can see I shouldn't be in an AI discussion. Okay, so I'm here with my mQACC hat on. This is a community-led initiative to promote the development, dissemination, and harmonization of best quality assurance and quality control practices and reporting in untargeted metabolomics research. You have to have good lungs to say that in one breath. Okay, but it's a fantastic bottom-up initiative. Its basic premise is that we're a community of volunteers and we focus on untargeted metabolomics. And the important thing here is everybody involved in mQACC is an expert, and they're an expert in quality assurance, quality control, developing new things, and we get together and we're really working as a community to try and improve mQACC, but most of all metabolomics for everybody. It's open. If you're interested in joining and you think you're interested, come and discuss it with me and we can discuss membership requirements. They're really difficult. No, they're not really, but come and discuss. So like Rick said, we're very much into persuasion rather than enforcement, and we do a lot with training and discussion rather than being the metabolomics police. Above all, we're standing for transparency of approach. Know what you've done, be clear about what you've done, report what you've done, be honest about how good this makes your data and where the potential failings in your data may be, what biases there are and how this affects your work. And we do a lot on risk management and how this may actually improve processes over time. So I've discussed our mission and we've divided that into 4 objectives. We can we're trying to identify, catalog, harmonize, and disseminate QA/QC best practices. We're looking at establishing mechanisms that the community can adopt them. We're looking a lot into training of best practices for the community, and we're encouraging prioritization and development of reference materials. And to actually get there, we have a lot of bottom-up working groups being developed. If you have your own ideas in mQACC, you can actually suggest them and they go through a committee to be approved. And we have LCMS best practices and living guidance. That's, if you like, our prototype technology group that acts as the seed for other technologies to be able to use their structured framework to develop things for their own technology. Quality assurance, reporting standards, reference and test materials, and community engagement. And then we have specific working groups on specific technologies for NMR and GCMS, and a literature survey group that does a lot of great things with literature reviews. And I'm not going to go through this in detail, but I will say that it's a lot about how we can improve practices, what we need to report, and adapting to new technologies. The major contributions that mQACC is attempting to make for data harmonization is mostly about defining what's reported so the data can be reused, and defining best practices so the data is collected well in the first place. And this development of reference materials is super important. Literature reviews, community engagement and training, and expert group for networking. That's actually a major benefit of mQACC. So I want to introduce you to our best practices living guidance document, and if you're interested in this, go over to Southall 2 immediately after this workshop where you can learn more about it. There's a workshop on it. They are looking at 8 different areas. The things in orange are those that they've already got living guidance on, and the data quality and quality assurance should hopefully be done by this autumn. And it's living, which means that this is a document that will be updated and changed over time. I'm here with my co-chair reporting standards hat on. We looked at reporting standards specifically for the analytical side of things, and we published this paper. And we went for a very simple template, CSV or Excel. In retrospect, machine readability may have been a good idea, but one of the advantages of this is that you can print this out and stick it in your lab book, and we've tried to make it as accessible as possible for all. And I'm confident that ChatGTP can perhaps solve some of the machine readability issues. We'll see. We've covered 11 different categories, including specific technology-related categories for MS and NMR, and we've included a glossary. And we have this idea of rapid reporting. If you don't make it easy to report, people don't do it. So the idea is you set this template up once, and then if you're running the same method again, it's then very, very quick to change it for the next thing that you do. And this template can then be printed out and submitted as your quality control summary to any journal you submit to. So we cover a lot of the different quality control samples, and people always think of pool QCs, but quality control samples include blanks, system suitability tests, replicate analyses, reference materials, and platform-specific samples. And this allows us very much to be able to judge and use your data in future projects. If you have blanks, you know what's not in your sample, but it's coming from the blank. If you have reference materials, you can match them up with other data. And if we look at enforcement, we've been very successful in discussing with editors the idea of including this 8-question submission questionnaire. So Metabolomics and Nature Metabolism have now included this. I have to acknowledge Rick Dunn, who led this effort in the community engagement group. Reference and test materials is being led by Raquel Cumeras and Clay Davis. If you're interested in this survey, they're looking at NIST 1950. Let's discuss the challenges. The original quality reporting guidelines were first printed in 2007, and Rick's already discussed how few people have adopted them. We suspect this is because they're quite complex. They require real intensive thought to actually get the data down on paper or in your computer, labor intensive and no reward. We require different reporting standards for different instrumentation, and different subsets of metabolomics or small molecule analysis may require different strategies or levels of rigor. If you're doing a pilot project of a new technology, what you need to report may be very different to if you're doing a clinical study with the idea of clinical translation into the medical field. So we need to bear this in mind as we're discussing reporting standards, and we need to make this as easy as possible. And one of the things that we're trying to do is actually discuss this with instrument companies and software developers, that they do a lot of the hard work for you, and by producing easy-to-read reports on methods and parameters, we take a lot of the work out of it. We also had a lot of discussion on equality and justice and not making things so onerous that we're pricing new labs out of the market. And this is one of the reasons that while we think reference materials are a good idea, we don't want to mandate their use, but we would recommend think about your benchmark data set that other people can use. And how we communicate this well to the non-experts. And non-experts in this case include general public, but they also include our funders and often the politicians making the more strategic scientific planning, because this will require funding. If there are any funding bodies and publishers interested in discussing this further, please come and chat. And finally, because when we were discussing this, we were discussing lessons that we'd learned from our own lab. This is less AI and more analytical chemistry. We've done some pretty large, well, I'm told they're not large, but we've done some sort of mid-thousand sample data sets now. And this requires a special sort of planning for this data to be reused. And so I've put this really for the discussion, that what we've found works best is when you start with clear objectives and scope, and you have a very clear plan. And the longer time you spend planning, often the easier the study runs. And then the three really important things that we've found is transparent and accurate reporting within the lab, because this helps with these large studies, ongoing mQACC, and harmonized terminology. And this one I really want to point out, the harmonizing terminology, just from an analytical perspective, we've had discussions on what 1 to 3 actually means as a ratio. And that's just the difference between analytical chemists trained in different countries. So be really clear with people what your terminology means. And we're all in agreement that you can't do data merging without having these 4 pillars of fair data storage, a failure culture in the lab. And by failure culture, I mean if I stand up and say, oops, sorry, I ran the wrong sample. Who hasn't done that? You need to be able to say that and know that your boss isn't going to shout at you and you'll still be respected by the rest of the lab. And in fact, I take the attitude that if I haven't heard of someone's failure in the lab within a year, then either they're exceptionally good or we need to be having a discussion about being honest about failure. And that can be, that's also a question about international ways of communication and finding a good way of communicating. We need realistic personnel and deadlines and funding, because the classic thing is we're short-staffed, we don't have enough money, and we promise to do something in a year that takes 3 years. And I'm looking at Thomas Hankemeier now. We need really good statistical support and training, and not only for those doing the AI, but also for those feeding into the AI, because we need to understand what you need at the other end. Lastly, if you're interested in quantification, then you may be interested in our latest mQACC survey, and I have 39 seconds left for you to look at the QR code. I'll give anyone who's interested time to do that.
Fantastic, Jennifer. Important. So I hope the audience now is seeing a series of presentations that can help you decide, where can I contribute? Where do I need to be a member of? And we want to hear from you. Please call, contact any of the speakers, any of the initiatives that you see being presented. And we either can take one or two quick questions, or we can keep moving and keep the podium, keep 20 minutes for a discussion. I'm in favor of the latter. Let's keep moving then.
I think David had a question.
David had a question. Sorry, here we go. All right. Peter and David, yes. Here you go.
So great presentation, and you're doing great work as a group. I'm curious about why mQACC doesn't want to include targeted metabolomics in its mandate.
So this is actually a question that we've had a lot of internal discussion in mQACC, and I think, you'll see by the last survey that with quantitative metabolomics, we're kind of edging that way. But the current status is we have limited time, and until we've solved the untargeted, we don't necessarily have full bandwidth to be taking on the targeted. This is not that we're against targeted. It's more about making sure that we can deliver on what we've committed to.
Okay, great.
And I think that there is a good groundswell of people in mQACC who would like to do targeted, including myself. So this is definitely on our radar.
Thanks, Jennifer. Just very quickly, I didn't see anything on your sort of range of topics around missing data, and I just wonder whether that's on your radar in terms of missingness and how and when that should be reported.
So missing data we were concentrating very much on QCs, and our current approach with QCs is we're often using the QCs to filter out missing data. However, many of us are now actually using the missing data to analyze separately, both from a technology point of view to see where our technology may have weaknesses, but also from a biological point of view to actually see what that missing data may be telling us. And class-specific QCs is something that we're definitely going towards to try and solve that. That's a whole lecture in itself. I agree with you, it's really important. And you've also reminded me, I even made a note in my hand, IDs. IDs, and this is a massive challenge with data analysis when you don't have full IDs. I need to get off the stage before Rima throws me off.
Thank you, Jennifer. Xianlin, and thank you. Okay. Han, a perspective of the lipidomics community and their effort in coming up with minimal standards.
All right. Thank you, Rima. So after the first talks, after Rick and Jennifer's talk, probably people are kind of wondering. It's already, it's a metabolomics standard initiative as well as quality control. So also everything is moving on very well. And for almost 20 years now, the initiative Metabolomics. So why need a Lipidomics standards initiative? So that's kind of leading me to give you a little bit of introduction of how we initiated the Lipidomics standard initiative. So here is what's going on. Is 2015 is almost 10 years later, the Metabolomics standard initiative. People are using the targeted metabolomics approach or lipidomics approach, and defines try to understand under the lipids is whether it can serve as a biomarker, like the paper published in clinical chemistry, and found there's two classes of lipids. That's PC and the lyso-PC, and some species is very specifically it's identified and negatively correlated with the development of type 2 diabetes. And then if close look, a few of the scientists listed the name, the letter to the general editor, and found is this lipids identification is very off. And there's a way, it's really is the species yes. And even after came back from the authors, correct what the species could be, and listed on the bottom. I even at this moment, I don't think that's even right. Okay, so that's kind of the okay. The citation of this paper still is very big. Even the last year is still is kind of citing as such a species as a potential or as a biomarker. And the 2020 is another study is coming out using the very advanced mass spec technology and using AOC and ion mobility as well as the parallel fragmentation approach, and identified over thousands of lipid species and from minimum materials. And it turned out, if close look, and about 500 people is questioned, the lipid species is questionable. And listed here is the lysolipids, and they spend the fairly long retention time as well as different of the CCS, and then obviously is something is wrong. So is a group of scientists and wrote a letter to the Nature Communication and published. Seems likely annotation of lipids needs quality control. It's really very critical, and something needs to be done. And even that, and this year, it's a paper published in the Science Advance, and a very nice work using the very sophisticated and the method called NMOST, and combines all kind of advanced technology, and using the analyzing both the proteins and the lipids, and they did the correlation, and it turned out some protein change correlated very well with a few called DAG, alkynyl DAG species. DAG means the vinyl is bound on the SN1 position, and is usually associated with a class of lipids called plasminogen. And when close look at this study, and it turned out that this molecular species, even the M over Z mass, is not matched well with the structure. And it's more than that. And if you look at their data, and again, just like the last paper I mentioned, and there's a lot of problems with the retention time and the eye mobility parameter correlation, and really is heavily problematic. So this all kind of things making the community, the people studying the lipids, the questioning is what are we going to do for the lipids? Clearly, the lipids is very different from the aqueous phase metabolites, and it's much more complicated, and the quantity. Not saying the quantification, even the identification is still have some, it's concerns and very challenging. So that's kind of leading to the Lipidomics standards initiative around the 18, the 18, and then that moved to further leading to the establishment of the International Lipidomics Society. So let me give you a little bit of this society. This society is, of course, kind of the members from the entire overall the globe, globally. And even the board members is from the 4 constitutions. And you can see it's like the Peter Meikle and the Aaron Baker will give the plenary talks in at this conference. So that's kind of the Lipidomics community, it's covering. And of course, the society is mainly one of the first development of technology, techniques, and the methodology, resource, and whatever, everything to motivate, to promote the lipidomics. And our core activity is mainly on the topic is today what we are saying. It's standardization, harmonization, and the establishes reference data. Okay. And all this, of course, the standardization, thanks to Rick, is mentioned about the minimum checklist. Actually, is now it's more detailed. The checklist version is published last year in General Lipid Research. And the harmonization is through the variety of the interesting groups I will mention a little bit later. And the reference data, of course, is many things, including the ceramide, which is already published in Nature Communication, and the other class of lipids, particularly in the plasma, is kind of ongoing. So, as I mentioned, the main activity or main effort is through the interesting group. So we have over 10 interesting groups in the society. And at least 5 of the interesting group are doing the link trials for the related to the interesting topic, like the clinical plants, the microbiota related lipids, as well as the references, as I mentioned. So let me give you a little bit of the LSI interesting group. It's already reporting the minimum report is published in 2022, and so it's after 3 years, more it's kind of around 3 years. So here is what's the outcome. It's a Rima requested to say is what's going on. So so lots of users is kind of being using the minimum report, and already have associated with 44 publications and reporting the study data. And in this minimum report, all the aspects of the lipid analysis being included. I don't have time to walk you through every, its a component. I just quickly go to particularly more or less, related to the, related to the how to make a relatively the more accurate annotation of the lipids. So it's all kind of content, it's from the mass, or it's the isotope effect, or the solvent condition, or the mass spec, the negative ion, positive or negative ion mode. All these kind of aspects are being considered to determine whether this class of lipids is belong to this class. So that's kind of, at least when you do the report, and you can give you some kind of idea, say it's whether the class of lipids you are reporting is correct. Okay, so I do not say do not have time to go through, and that's you can do everything is it's there, and of course regarding the development of this initiative, so we are adding the imaging, adding the GC mass, adding the untargeted lipidomics approach, and to the all the workflow, and also actually including the GC workflow, and the many other activities is going to be developed. Yeah, and so different interesting group, and particularly I want to mention about the this one, the oxylipin analysis one is published, the guideline recommended, and over 100 authors is co-authored, and indicating the endorsement. Okay, I will stop here. That's actually my more like personal comments for on the lipids. I do not have time. Thank you.
Han, we want to make sure the communities connect. Lipidomics, metabolomics, we are studying metabolism, and therefore every single effort to connect all of these activities is critical, is a must. So I'm happy to see we are getting together, back together as we should, metabolomics, lipidomics, and so on. All right, we're gonna move on next to Peter Meikle and talk about data harmonization. Right, we wanna make our way at the end to the, 10 minutes would be great.
It's okay, thank you, Rima. So I've been asked to talk about harmonization of lipidomics data sets, which I think is so critical at this time. There's just a tremendous amount of data out there from many large studies over the last 10 or so years. But we're not really using those data effectively. We're not bringing them together. And so that's really what I'd like to address today. Of course, why would we want to bring these different data sets together? There's a number of reasons, but one obviously is power. If we can bring different data sets together, perform meta-analyses, then we increase our power and we can see deeper into the lipidome. And this is one example from Rima and my lab's work to look at Alzheimer's disease and doing a meta-analysis of two quite large cohorts. The other area, of course, as we start to move into genomics and genome-wide association analyses, it becomes even more critical. So having very large data sets of 10 or 20 or larger numbers will be essential if we really want to dig deeply into the genome-lipidome associations. Okay, the other area really is, and I'm sure most people who have a lipidomics platform would recognize, if you compare your platform today with what you were doing 5 or 10 years ago, you've got an enormously larger platform today. So we've gone from about 350 lipids now to 800 lipids. And so does that mean all the analyses you did 5 or 10 years ago suddenly aren't really that relevant because now you're looking at this broader platform? And of course the answer is no. And this paper we brought out just last year addresses that issue. And essentially what we can do now is take that smaller data set and impute all the missing lipids from our larger data set. And this works really well, and the reason it works well is because of this high correlation structure that exists particularly within the lipidome or the plasma lipidome. So every lipid within plasma is highly correlated to many other lipids within plasma. And so using a series of regression analyses, we can predict or impute all of those missing lipids, and this is what the paper was demonstrating. And so, you know, for probably a 90% of, in this case, we were missing over 400 lipids, we could impute over 90% of those lipids with an R-squared greater than 0.8. And so that really then allowed us to do comparative analysis across a cohort that we ran 10 years ago with one that we run today. Of course, that's in the one platform. The real challenge, I think, is to start to go across platforms. And I think everyone would appreciate that Biocrates and Metabolon have analysed many large cohorts over the last 10 years. And so what started as a small ring trial then turned into this objective of trying to harmonise across different platforms, and particularly across the platforms where we've got large amounts of data. And so we were looking in these examples, I'll show you, really at Biocrates, at Metabolon, and at our lipidomic platform that we developed at the Baker. And one of the main challenges here, Han alluded to this, I think, is isomeric and isobaric species. And so different platforms measure those species at different levels of resolution. So in the Biocrates platform, they measure this PE acyl alkene or acyl ether lipids, in this case C36-2, as a single unit. In our platform, we're measuring these, I'm not sure if I can get the pointer to work, we're measuring these as multiple isomers, so PCO 36-2 and PCP 36-1, as well as the diacyl form PC 35-2. They all come out as the same, within the same MRM transition, but you can separate them chromatographically. And so while we're measuring these as individual species, some other platforms are measuring them as a single entity. And so that makes, again, comparison of different platforms very difficult. Kevin Hun in my group came up with this approach of really adapting from our imputation of lipids, but now adapting across platforms. So we take in this case, our Baker Lipidomics platform, we create multivariate models to predict the Biocrates platform, so the PCAE36-2 that they're measuring. And so we use lasso regression models to do that, and we get a number of lipid species contribute to those platforms. And so now we can see quite good correlation, good agreement across these two platforms. Of course, that's fine because we're going from a more resolved platform to a less resolved platform. The question really is, can we go the other way? So can we use Biocrates to impute our highly resolved platform and all of these individual lipid species shown down here? And of course, the answer is that we can. So now we're taking the lipid species in the Biocrates platform, and we're using those to create models to impute the Baker lipidomics, in this case, the PCO 18.0/18.2. And again, here you can see we actually get very good correlation between our imputed lipids and the actual measured lipids on our platform. Okay, so that's the sort of the framework, I guess, that we're working in. More recently, we've gone on to do the same thing with the Metabolon platform and created a whole series of algorithms that allow us to impute back and forth between our lipidomic platform and the Metabolon platform. And so in this particular example, we've again used lasso within 10-fold cross-validation to predict the Baker lipidomics using the Metabolon platform. And we had a training data set based on the Tulsa study, about 774 individuals, 3/4 female, quite a young age. This was a depression study. And then we've got a second study we use for validation, the ROSMAP study, smaller sample size, same percentage female, but a much older study. And so we imputed from Metabolon into our lipidomic platform. And what you can see in this panel here is that the internal validation in the Tulsa works quite well. Again, probably 80 or 90% of the lipids have an R squared above 0.75. And again, this is very similar when we take the models created in Tulsa and apply them to the ROSMAP data. So these models do work across when we cross-validate into an independent cohort. And the models are actually surprisingly quite large. They're lasso-based models, but they typically use between 50 and 250 lipid species. Most of those lipids will have very low coefficients, and typically there'll be a number that have a high proportion, but it does, I think, support the models having these large number of lipids within those models. And so here's just some of the data that we're generating with this approach. So here, this is all internally cross-validated within the Tulsa. So we've got our measured lipids in the Tulsa, and we've just looked at the association with sex. That's on the left-hand side there. This was with our measured lipids. And then within the Tulsa, we imputed our lipids from the Metabolon data, and then we do the same association with sex with those imputed lipids. And this is just a plot then of the alignment of the beta coefficients of each of our lipid species against sex. And you can see the imputed data gives pretty much exactly the same answer as our actually measured data. And again, another example looking in the same cohort, looking at depression as an outcome. Again, you can see these, really highly correlated profiles, and shown here with the beta coefficients. And particularly we're interested in this ether lipid, phosphatidylethanolamin, and then the ether lipids of ethanolamine, which show this really characteristic, which we've seen in a number of cohorts now, but are very strong in depression. Okay, that's all internal validation in a single cohort. We also wanted to look at, could we do that across cohorts? So create the models in the Tulsa, and then apply those models into the ROSMAP data. And again, this is looking at the association with sex, and again, our measured data in Tulsa, and now our measured data in ROSMAP, now our imputed data in ROSMAP, using models created in the Tulsa, again giving very similar profiles, and again highlighted here with the alignment of the beta coefficients. And finally, the same effect if we're looking at depression in the ROSMAP. This is, it's really even a slightly different measure of depression. In ROSMAP, it's based on a questionnaire. In Tulsa, it was based on clinical diagnosis of depression. But again, we see these profiles agree very much. Again, the correlation not as strong as internal cross-validation, but still very strong. And we can still see this characteristic ether lipid signature probably even more strongly in the imputed sample than we do in the measured samples. So in closing then, there's this very strong correlation structure that exists within plasma lipids, and that allows us to do a number of things. We can impute missing lipids, we can impute lipids across different lipidomics platforms, and we can help define these isomeric and isobaric species. And of course, the important thing, the imputation captures a large portion of the variance, typically 80% or more of the variance. The caveat I would say though is that it doesn't capture 100% of the variance. And so most of the analyses we've done, we see very good agreement, but of course that missing variance, if that missing variance holds the variance for your association that you're looking for, then you could miss that. And so we need to at least be aware of that and be looking for that in these types of analyses. But the benefits, I think, far outweigh the potential downsides of this, because it really does allow us now to use large amounts of data that have been acquired over the last 10 years and really dig much deeper into those associations. This, of course, has been done under the sort of auspices of the Alzheimer's Disease Metabolomics Consortium in the US, and there are many groups involved in that, and so we've enjoyed working with all of those. I'll stop there.
Great. Thank you. Thank you, Peter. Thank you.
1 minute.
So, I hope this illustrates the power of taking big data to knowledge. All of these studies, Peter, whether it's a depression or a brain, Alzheimer's, or a blood or anxiety, we are trying to connect this data to learn about the brain. What you are developing as foundational work is going to be essential for all of these initiatives funded by NIA. I just want to acknowledge the National Institute on Aging that is enabling us to put data in real time, make it public, correct it in real time. We understand that the data is not fixed. The beauty about the thinking, you put your data out there, you have a podium to correct it, update it as you learn, which is critical. And the ability to connect this data to derive knowledge, I think, is key. We want to add the component of the exposome. I think Martin is next. Thank you, Peter. Okay, let me see. I think, so if we see things that do change in a disease, we want to understand, is this the genome, is it the gut microbiome, or is it the exposome at large? We want to take a snippet into the exposome, the complexity of the exposome, and the integration of data at the end, the larger metabolomics data with the exposomics data, to learn about the influences of the exposome.
Hello, so it's very nice to be here. My name is Martin. I am here on behalf of Emma Szymanski. I'm not sure if I know how the pointer looks.
The screen here, so you can.
Oh yeah, I see now. So I'm not her. Emma is my PI, but I will do my best to represent her. But please be nice. So I have a few slides on beginning when I will talk about exposomics. So exposomics, what does we mean by this? So this is the further downstream omics, which is closest to the disease as possible. So for us, it's very interesting area of the chemicals which we want to explore. But because it's so far from genome, the complexity increases a lot. So we talk usually about millions of the chemicals which we look for. There is just another, the definition from the 2014, where we look at the exposomics, we look at cumulative, the measure of environmental and associated biological responses. So we look at the both environment and response of the body to it. And this can include also the diet, behavior, and the another endogenous processes. So when we talk about exposomics, we have very vast space. So if we look at the PubChem, you can find 119 million of the chemicals which we look for. Unfortunately, we have MS2 for only 0.008% of the chemicals, so very low amount, very hard to assign the name. So if we compare exposomics with the classical, let's say, metabolomics, you have two ways how you can run the samples. One is for the targeted, the analysis, where we look at the subset of known the compounds. Usually we have the standards for them. Or you can take a look at the non-targeted screening, where you look at much as possible in terms of extraction procedure and the sample. So if you do this in the exposomics, what you usually get, so you use the databases which are available, which is the PubChem CAS, where you have millions and millions of the chemicals. What you usually get is the thousands of hits for each individual MS2 the spectra. Because like I said, we don't have MS2 in the PubChem, so what we do, we usually predict it in silico based on the structure, which leads to the thousands of hits. So this is example of the nicotine, and you can see we have almost 3K of hits for the nicotine itself only. But only few of them have some informational content in the PubChem itself. Most of them are the candidates which don't tell us too much about the chemicals themselves or about the samples. And in addition, the runtime, it's very long. So what we try to do, we usually in Exposomics define something which we call the suspect list, which is predefined list of the chemicals which we look for in the sample. For this, we built the databases. One of them, which I will talk about a bit, is the PubChem Lite. So the PubChem Lite is pre-built from the PubChem. In the PubChem, if you look at the table of the content, you can see the different categories here. And if you look at them, you will actually find out that some of them can be associated with exposomics, like the pesticides, or there are the compounds which are connected with the food. So we started in 2020, I think, with those 8 TOCs, and then we expand them with the categories which are connected with diseases or with the biological pathways. And most recently, we also expanded with the chemical classes. The chemical classes include also endocrine disruptors and cosmetics, for example. So in total, we have 11 the categories which we filtered from the PubChem and built into the database, which is available for you to use. It's built each month. It's on Zenodo, and it can be used directly with any tools which can use this in silico fragmentation, like, for example, MetFrag. In addition, what we have done is to build the web page where you can actually search throughout this subset of the chemicals. So you can search by name or by the formula, and you have all the structure information of the compound. You have also related CIDs and also the presence of the compound in each individual list. This can help to define if the compound which you found is a drug or food, or if it comes from somewhere else. And in addition, we have also visual representation of the references. This can help quite a lot. If you find, for example, the drug which is not used, you will see that the references are actually quite old, not the recent one. And this can help you with actually realizing that the hit which you have may not be real if the compound are from the recent the samples. So what we aim for, we talk about the big the data. We actually aim for shrinking it down a bit for the Exposomics, where we can have the meaningful hits, less amount of hits, but the one which have meaning, and we can assign some type of information behind them. Yes, so these are not the only tools which we built in Emma's lab. I have still a bit more time, so I would like to mention also shiny TPs a bit more. Because when we talk about the Exposomics, we also talk about transformation. So what we built is that we took a chemical global inventory, and we predicted transformation based on bacterial and abiotic lysis using biotransformer 4. And we actually also built the web page where you can very easily search for the transformation of the chemicals which you have, or you can try to find the precursor if you have some hits from the Exposomics. Yeah, and I was a little bit faster, but that's my talk, and I think it's good. Rima will be glad.
Thank you.
And yeah, thank you for the time.
So Martin, we want to bring the universe of the exposome, the metabolome. Not only do we want to be able to identify these things that are in the environment, the chemical exposures, or gut metabolites, or drugs, or supplements we take, or the food, I don't know, millions, thousands, whatever number of chemicals end up being part of the system. In studying a human disease, we want to bring this universe together so we can understand what affects a disease. So all of the resources you are building must connect and crosstalk with the metabolome so we can study their influences on metabolism. Yes. I hope that is the ultimate goal. Yeah, maybe we can go to Tuulia. I don't know if the order, but Tuulia, a few minutes on applying applications for the study of the exposome, and then we move to Thomas, leaving 20 minutes as we promised, for a discussion on the future. Thank you.
Okay, so I'm showing with some practical examples how we combine exposomics data and metabolomics data together, and some of the critical parameters which you should consider on that. So we heard about the exposome approach. So basically, we are exposed to a lot of chemicals. It's estimated that at least 700 chemicals can be measured. This is probably underestimation. And there's like this dark metabolome. It's said that maybe a large part of it is actually environmental chemicals, the transformation products, and metabolites. A lot of these are classified as endocrine disrupting chemicals, which means that they can mimic, block, or disrupt the natural hormone system. So it's not weird then that they have actually a lot of adverse effects. Here we have been summarizing some of the most known chemicals and their health effects on the chemical pathway level, biochemical pathway level. And you can see in different organs they are a bit different. Oxidative stress is a common element, but then also a lot of lipid-related pathways are affected by several of these most toxic chemicals. Of course, this is not a complete picture. We have a lot of chemicals that we actually know very poorly about their health effects, and also even these well-known toxins are not fully characterized related to their health effects. There's a few studies what we have been doing. So we have been looking at prenatal exposures during fetal development, early life, showing that especially lipid changes are happening, and also we show there's actually gene-environment interaction. We show that there's sex-specific impact later in life. PFAS, for example, also modify the breast milk composition, also PCBs. That can, of course, have an effect later in life on the infants. We also have been looking at this kind of indirect effects via the gut microbiota, and also importantly looking that a lot of these chemicals have non-monotic metabolic responses, which means that you have to take this into account when you do data analysis and data integration. I take an example of the PFAS, perfluorinated alkyl substances, because we are all exposed to this. They are widely used, and because they resemble natural fatty acids, like you see the octanoic acid here. So the PFOS and P4, perfluorooctanoic sulfonic acid and perfluorooctanoic acid, resemble them in the structure. So, and as I said, 99% of human populations are exposed to these. And because they are looking at the fatty acids, so they are actually really well absorbed and really slowly eliminated in the body. And they have been showing a lot of different adverse health effects, which you can see here. Basically, what we have been looking a lot is like both the direct effects, but also the interplay between PFAS, bile acids, lipids, and gut microbiota. So bile acids are very important metabolites because they are acting as signaling molecules controlling glucose, lipid, and energy metabolism. And of course, they do have also important role in lipid digestion and absorption after the meal. But here, the important thing is that they actually, PFAS can disturb the bile acid metabolism and the whole enterohepatic circulation. So they can suppress the bile acid synthesis in the liver. They can have an impact on the bile acid conjugation in the liver by downregulating this HNF4. And importantly, they are also modifying the gut microbiota, which changes the secondary bile acid synthesis in the gut. And a lot of these secondary bile acids are actually important signaling molecules controlling the glucose and lipid metabolism. And then they also compete with the transport of the transporters of the bile acid when taken back into the portal vein and back to the liver. And then in addition to this, PFAS has also actually direct impact on lipid metabolism, for example, fatty acid beta oxidation. And showing now a little bit of examples, this is also like important factors to think on data analysis. So we did liver biopsies, matching serum samples, then combined it with animal studies. We did a comprehensive metabolic characterization, lipidomics, also targeted methods, and then we did a global profiling of the exposome. And what we did find out, this is important finding, is that you can see here, let me see, does it work like this? No. Yeah. So anyway, you can see males and females here, and these are bile acids and lipids in the liver, and you can see that they are opposite patterns. So these are fold changes against the controls. So you can see here that the males, actually, the PFAS are suppressing the lipids, and this is probably due to the suppression of this CYP7A1, the bile acid synthesis enzyme. While in the females, we actually see opposite, and we see also same time increased level of this kind of lipotoxic lipids like triglycerides with short saturated fatty acid in their structure. And especially this ursodeoxycholic acid, which is the secondary bile acids, which is actually shown to increase the expression of this CYP7A1, and this leads to enhanced bile acid synthesis, and this could potentially explain the sex-specific effects what we have seen, that PFAS and this UDCA are competing of this CYP7A1, and that is important because it can explain what we see, and this is really a gut microbiota-related metabolite which we see. And basically, this is also, UDCA has been actually used for treatment of patients with fatty liver disease, and it has been shown that in obese subjects is actually increasing the hepatic triglyceride formation, so which could actually explain what we see here. So we see increased level of UDCA, and we see increased level of these triglycerides in the liver. And this is then also connected to actually the health effects. So you can see males and females here. We have also here other compounds, but looking at the PFAS only. So PFAS is associated with increased liver fat, increased HOMA-IR in the females, but in males it's actually associated with the lower liver macrofat, so showing that it should, in these low levels, have a kind of protective effect in males. So this is very important finding, so that we can't just adjust for the sex in these studies, because if you have opposite effects and you adjust, so you will lose these important findings. And we see also these monotonic effects. This is on 2 different hepatocytes we have been looking. So polar metabolites, lipids, with the PFAS levels increasing from left to the right. You see also fold changes against the control. So in the lower levels, we have increased level of a lot of polar metabolites and lipids, and then downregulation at higher levels. And this is what we see also on humans. This is two groups of occupationally exposed aviation firefighters, with the senior ones having high exposure levels and the more junior ones lower exposure levels, and again, correlation with the PFAS, and you see totally opposite patterns, like in a lower level, it's increased levels of a lot of these metabolites, and at the high exposure levels in the upper panel, it's like a negative association, and as you can see, if we put it on here, so it follows very well what we see in the hepatocytes models. But of course, hepatocytes, you can't get this whole organism level, so we have been then looking at also the gut microbiota associated studies, and here again, not going into detail, the main finding is there's a lot of these compounds are associated with gut microbiota in a sex-specific way, especially this Prevotella copri, which is associated with PFAS in females but not in the males. And this is important finding because this Prevotella copri, we have been finding in our previous studies, is actually associated with insulin resistance in this kind of obese cohort. And this could explain some of the findings. And basically it's associated also with the biosynthesis of branched-chain amino acids. But importantly, we also saw in the same study, not going into detail again, that actually these changes on the gut microbiota are associated with changes in secondary bile acids, which are actually mediating a lot of these health-related changes, like UDCA, which I was mentioning earlier, but also lipocholic acid. So basically, that a lot of the changes we see are indirect effects via the gut microbiota and the metabolites the gut microbiota are producing. Then just a final one, this is because I wanted to show that actually how metabolomics can actually really use as a sensitive readout of adverse biological effects. These are eye drops commercially available, which are 100% PFAS. We can buy it then without prescription in Nordic countries, and it's very poorly characterized on levels of toxicology, so there's no proper ADME studies. So we made a quick experiments of the hepatocytes, and you can see the dose coming 1 to 28, so we have these are fold changes, and these are significant ones on lipids and then polar metabolites. So we really see non-monotic response, which we saw also with the other PFAS. We see a major changes on very key metabolic pathways related to fatty acid, amino acid metabolism, sphingolipid metabolism, and pathways related to energy metabolism. So basically they are suggesting that we see mitochondrial dysfunction, increased oxidative stress, and indication of also, we can also see that actually this compound seems to be metabolizing into the liver, which is actually not surprising because similar compounds have been showing not only to be enriched in the liver, but also metabolizing. So this is like a very fresh study, so showing that, yeah, the metabolomics is actually a great tool looking at this kind of adverse health effects. And with that, I'm just finalizing. So basically, looking at the PFAS, but other compounds also, we see clear interactions. We see them early life, but we have to take into account the gut microbiota because they are also modifying a lot of these effects. And then they are like clear sex-specific effects, and this has to be taken into account on the data analysis, and not just adjusting the models, but really looking the effects separately, and also because most of the study results are not linearly like correlating, but there's monotonic effect, so you have to use a little bit different approaches for the data analysis. And yes, and then also the gene-environment interaction is very important part and should not be forgotten when looking at the results. And with that, I'm done.
Fantastic exemplification, Tuulia, of connecting then the chemical exposome, the gut microbiome, the metabolome, connecting this big data to understand contributions to human health and disease. Fantastic model. We move on to Dr. Thomas Hankemeier, and everybody has been patient. Thank you. I look towards the future. Of course, we want to make the data usable. We want to make it machine readable. We want to make it available for AI. Everybody is moving to applications of AI, so are we.
Good. Thank you. So I want to talk today about FAIR data, and as you may know, the FAIR principles have been launched in Leiden in a workshop 11 years ago, and in that FAIR was interoperable already at the core. So that means that in principle FAIR data should be anyway machine readable. And if FAIR data are machine readable, they are also ready for AI. And that's what I want to elaborate a little bit more on it today. And I also, let's say, I'm from Leiden University, but I'm also co-founder of the Leiden Initiative for Fair and Equitable Sciences. And I want to mention here some co-workers, Baran Mons, Erik Schultes, Ari Bach, and Tilda Baumann, but also many others who worked on this now for many, many years together. And this is not only us, but it's a very big consortium, public, private, in the Netherlands, but also very much worldwide, linked with many, many other initiatives. Because during the workshop which we had a year ago, where we celebrated 10 years of FAIR, it was very clear that, and not only it's us to promote it, but many agreed, that maybe we should stop with data sharing and move to data visiting. And then you can say, what, why? Well, actually within, let's say, our LIFES, we are not only looking at metabolomics mass spec data, but we also look on health. For example, if you look at mental health, the financial situation is actually one of the key determinators whether someone has good mental health or not. These data are not downloadable at all. Let's say in the Netherlands, I'm German, but the Netherlands is very good organized. All data are available, but of course not downloadable. They are at the Central Bureau of Statistics. So you can search there a lot. You can also look where people live and what the air quality is, but again, as all the data are very much interesting connectable, that's not what we want. And actually we have companies involved in LIFES which are also organizing when I do my tax, let's say, request to get the return. I guess you all do that. It's fun enough what they find. Not, let's say, actually they found my bank account, which I had lost when I was in Germany. It was really amazing from 20 years ago. And the same is for the health records. In the Netherlands, we have the Health Research Initiative. And when I, let's say, look for all my health data, the algorithms are just searching over all different resources in the Netherlands, and then they find where I was once at the doctor, and there are some data. So the system works. And this is not only for health. Within LIFES, we are also looking, obviously very interesting, this is for the police to trace people and to collect all the information. So private sensitive data are very important. So how do we look and organize this? We have to actually link then data repositories, the infrastructure, computing infrastructure, but also algorithms all together. And that works if, for example, agents want to ask for certain data, then they come to a central point. There are defined libraries how agents should interact with the different repositories and resources and to check what via the different FAIR endpoints can be found in which repositories. But then there is also the question back to the agent, who are you? Are you, for example, a researcher? Are you a health insurance company, or maybe even life insurance companies? And that will very much determine, of course, the access. And again, that's not about mass spectra data now only, but this is also linking it with health data. So as I myself, I started with Metabolomics like Rima many, many years ago. But if you move to Exposomics, mental health, and all these things, you need much more, let's say, dedication on this if you want to link the data. But that's what we want to link. And also, that's also my personal aim, to make really metabolomics usable for every citizen and patient. If all these people will contribute their data, we have to orchestrate properly. So that's what we do within LIFES. So our principal starting point is the data should remain at the source where they're generated, but then being FAIR generated. So that these data are machine readable, that they are made visitable, and then data can be reused. That was also the idea of the FAIR principles. But then we take care that this is really done in a credible manner, which is also important because we want to have data from all over the world, and not someone should own then all the data. For example, if you look, we are working also with lots of people in Africa and Asia and other, let's say, continents together. If you look at data on infection, on other things, that's what we have to then take into account. If they want to share the data, maybe that won't work. But if we say, yes, but we want to only visit your data, then they're very interested. So actually the first concepts of these FAIR principles were landing and established in Africa very successfully. But of course, privacy preserving, having the proper consent and the proper access is then very important. So this is how it is now, often, let's say, being evolving. There are different standards, the data are created, data are harmonized, we have data warehouses so that you can ask the different questions for data reuse. So, which is all good, but what we then try to do is to have these FAIR, what we call FAIR endpoints, that then these data resources can be actually even linked among each other, using these minimal open standards for the representation, so that, let's say, all the different standards, of course, are good, no problem at all, and computers can translate very easily all the different standards as long as they are, let's say, well defined. So in such an ecosystem, we are convinced that this can really accelerate research a lot. An example, we in our own lab, we are generating a lot of metabolomics and exposomics data using mainly mass spec. The first reason why I moved to FAIR was that if I want to organize my own data properly, I can benefit a lot. So the first starting point for FAIR, my enthusiasm was, well, if I have a FAIR lab, I can learn much more about my data and can use AI, for example, to recognize what is a peak, what is working well, what is not working well. So, but if you want to do that, and that's exactly what, let's say, then makes data very much linkable, For example, we are using now nano publications to define all the different metadata, but then we can use our data with the data, for example, which Roel Vermeulen collects for the exposome maps in Utrecht, because then different endpoints can link among each other. We can link exposome data to metabolomics data, to mass spec data, exposomics data, but also even to the other information which is around in the Netherlands. Well, making metabolomics data fair is beyond going that you just have an Excel sheet where you define what is what. So the Netherlands has a lot of funding for open science. So we got there a program for 2 years. So to lead maybe by example, to work with fair implementation, to define a fair implementation profile, which all metabolomics group, at least in the Netherlands, accept. And that's what we are going to do in the project, Verify Metabolomics, to address, say, some of the bottlenecks which we encounter. And then if, well, the Netherlands is very small, then we can even meet in person very often to see what are now the issues which hamper the implementation of FAIR. And if that is all properly done, we are working on how to integrate then the data, or working, actually that's what we are already practicing in certain instances. For example, during COVID, we were able to look where are which patients treated how and what was the response. And then, for example, certain pharma companies, we could help them to find where are the right patients with the treatment and did it help. But this is also, for example, being developed now for side effects. Side effects are reported by, let's say, an organization in the Netherlands, and then you're able to trace who got which treatment, and then they get all this information. To finalize, if we properly have the data fair and make them data visitable, we can apply all our different AI tools, which, let's say, you, us, many others are developing and exploiting, and we have nice examples that actually LLMs can make data fair and then can analyze them even much more efficient. So there are many, many things, many companies working on this, which are all linked now together within these LIFEs, and there are many other initiatives now where they want to copy the idea which we had there to make, let's say, data fully AI ready, whether it's metabolomics, multi-omics data. We showcased already by ourselves that for our exposome that works, but I think there are many other opportunities, and if you want to link, we are very open to work together with other initiatives. Thank you.
Fantastic. Thank you. Thank you, Thomas. What? May I ask, just for the exchange, team, we would love to hear 2 minutes while we have the panel of discussants. Please move to the front. Mate, David, Susan, please, our discussants, if you can sit up at the podium here. While you get there, we know many people have questions and remarks and so on. I just wanted to give 1 minute to the exchange team. Fantastic presentations over the past 2 days. While you do this phenomenal effort to try to connect biorepositories, make the data more organized, usable, and so on, we are taking a perspective of making big data usable to inform about disease, connecting influences of the genome, the exposome, the gut microbiome, and so on. We must meet as communities, and the Metabolomics Society can play a critical role in enabling, promoting, getting ideas. I wanted to hear 1 minute from the exchange. Juan, anyone who can comment? Who do we have from? Yeah. Yes.
Okay, thank you very much, Rima. So just 1 minute, actually it connects very well with what just Thomas has explained. So together, Metabolomics Workbench, GMPS Massive, and Metabolize at ABI, we are really trying to standardize open data practices in the field. That means how data should be in public repositories, but much more than that. We want to really harmonize data standardization, at least for some key data types in the field that it also involves ontologies, and that connects very well with what Thomas has been explaining. Without standard formats and ontologies, data visitation becomes an exercise that can be only done by a small number of labs together. And this is really what we want to do in the next couple of years. It's funded by the Chance Hankemeier initiative, and I hope that it will have huge impact for the community. And it's also Lipidomics and Exposomics are included. They are not excluded, sorry.
We can find many ways in connecting these two initiatives that are certainly clearly complementary. And Rick, I hope we can get the Metabolomics Society to play a much more active role. We'll discuss in the town hall tonight. All right, so I leave the podium now with our discussants. We open the door. I'll just take questions. The
so I think we have 25 minutes time for discussions. So, my name is Mateo Resic. We have
Dave Wishart.
Susan Sumner.
So I think we didn't have very much time for questions, but I see that we've got a couple people standing up. So if you'd like to ask some questions, both to the speakers, and we'll try and facilitate comments.
Thank you so much for this wonderful workshop. A lot had been said about reportings, but not much said about the sample selections that can lead to a data, even if we have standardized reporting and instrumental, it's not necessary that it's comparable. We have recently project in different healthy controls, and when we use different healthy control in blood samples, we see different results, like about bacteria. It's a very good idea to use AI, especially on standardizing and cross-talking between metabolomics, proteomics, and all of these omics together. Especially when we use AI, is there any risk or what we should be careful when we run AI in some data that things can go not in, say that, what kind of factor that we need to really consider that validating the outcome. The data of metabolomics is quite different than, for example, single crystal data. As I said, sample selection can make a difference. Sample preparation can make a difference. Thank you so much.
That's a really interesting question. I think perhaps Thomas, if you wanted to maybe respond, because you've talked a little bit about AI and...
Yeah, well, wouldn't say that I'm the expert on AI. I have many friends, many of you can do that maybe even better. What I see and learned, you have to do it properly, not necessarily that AI answers are always correct. Hallucination is very big problem there. So that's also where we started with, if you use fair data and not maybe ChatGPT, so you have very correct defined sources, that helps. So then, for example, you can use a ChatGPT type of tool for your own data in the lab, and these tools are around. So having said that, so the data have to be good if you want to use AI. And for that, there are nice tools around. For example, Clara and many other tools are very nice to make your data fair. For example, if you have an Excel sheet, if you have a paper, then you can use these tools which are out there, and I didn't want to make any advertisement, so I didn't show the examples, but we tested several of them. They are very nice because then your tools are not only published as a paper, but then also as a fair data set, and then you can put them also, for example, and make that available. That doesn't mean that all things which AI does is correct. So all the normal cautions which you have to do, you still have to do. No. So the first starts, of course, having the right question, because if you don't ask the right question to AI, you get a very weird answer, what maybe you expect. And really, please use the right data sources.
Great, thanks. Are there other questions that people have? Yes.
So I want to ask...
Please, the podium, mics. Can you go to the mics?
Oh, yeah. So I wanted to ask the things which relates to it. So you wouldn't use ChatGPT or those tools to actually make the data fair then? Because there was the mention that this could be the tools which can help in this, in conversion, because we all have stacks of old CSV files and which the PhD left, and now basically it's very hard to make them fair on your own. But is there some way where we can use AI in this?
Let's say, for example, a very nice tool, again, is this Clara tool. You should check it. There you can just upload the data, they screen the data, and then they transfer it into something that also all the data, at least if they're ontologies, or what we just said, or Jan just said, if the ontologies are crap, it doesn't help. So the definitions have to be good, but the definitions don't have to fit exactly, for example, the current ontologies, because if you have tables, for example, where how you defined it and how that links to others and so on, then these tools can do that very much. And personally, but that was also mentioned in the presentation, and therefore I think mQACC does very good or very, very important work. But personally, I think fair data only makes sense if we have an error model for which we also report, because then you know which data to trust and not. Because just making all data fair is good, but if you don't know what is reliable, then it's less good. So therefore, I think that is very important, but there are very nice tools. So actually, some people use even ChatGPT to make data now fair, but I think there are better tools, but let's say there are different ones you can use.
Sorry, I just wanted to comment on that because I was the person who mentioned ChatGPT. It was partly tongue-in-cheek to use ChatGPT. I find it useful for some things, but terrible for hallucinations and false knowledge for others. But I do see where it may be helpful to use LLMs, so large language models, is some of the data wrangling that we have to do, where people have used misspellings or used a capital letter or a small letter when in some and the others have got capital letters. So that's all I really wanted to say. I wouldn't recommend ChatGPT.
Thank you all for a very, very exciting discussions about making the data AI ready. But I would like to ask the panelists to maybe wave magic wand and tell us what AI tools you would like to see that works well with your AI ready data.
So I think that's a really good question, and I think one of the points that's already been raised is that we do have a sort of a Tower of Babel with the data formats and data structures, and large language models, not just ChatGPT, but ones that have been specialized or fine-tuned to metabolomic data can be created, and they are being created now. And I think these things offer, I think, tremendous opportunities to make data more convertible, more readable, more consistent. I think it's also relevant, we talk about the raw data, but we're also ignoring all the papers that all of you publish, and that's where we're converting, data to knowledge. But that knowledge isn't accessible because it's sitting in a paper. And I think there's a real need in the community to develop tools that can mine the literature and connect it to all the raw data that you're depositing. And that way, that too becomes machine readable. So the conversion of papers, the knowledge, to machine readable components is just as important, I think, as the raw data and making it machine readable. And I guess we'll have to see how those things develop over the next few years. But then I'll give...
So just to clarify, I'm not interested in what technology to use. Everybody's going to use LLMs. But really, what is the magic wand? When you're sitting there analyzing your data, what do you want your tool to do? Do you want it to spit out, just take your data and spit out, here's insights, biological insights? So I'm really more looking of sort of the outcome of the tools that you're looking at.
Yeah, I'm not quite prepared to answer that, but maybe somebody else will. I did want to say that part of what's confusing to me about the AI ready part is all of the workbenches that require people to use the data on the workbench. So it's not downloadable and you can't harmonize it with other data. And so it seems to me like the air and fair is missing from those workbenches, like the All of Us workbench, which is run by the NIH, or the workbench for the ECHO program, which is the Environmental Influences on Childhood Outcomes. These require investigators to register on that workbench to use the data. So I don't really understand, even, you know, once you find it, it's the data is not something that you can pull out and harmonize and use it unless you're just working in that environment. So one of the things, if AI Ready could get us over this air and the fair being missing, that would be very helpful, but I don't know if it can.
I think we'll have to carry on to other questions. We can talk about some of these things afterwards. But.
Okay. Thank you for the talks. My question is a little bit not about metabolomics by itself, but you said that when you deposit the data, some of the data you can access, like it's public, and some of the data you need to request an access to. So what is being done to make sure that the data that is private remains private?
So I think Thomas may be in the best position about that. And maybe, Thomas, you can explain data visiting a little bit more for people, because it's a new term for a lot of us.
So maybe to explain how we do it, let's say now in the Netherlands. So of course, it's very difficult or complicated. Imagine that all our the health research infrastructure got 50 million to make health data which are collected at the different hospitals accessible to researchers. But how are you going to do that? But that's what is now being worked upon, and how do we foresee that? So the idea is that individuals can manage their own data. Now, this is now not about data repositories, mass spec data, but this is now who is the boss of the data. And that's also, let's say, coming back to the other discussion about publishers making data accessible and so on. That's all in the same kind of, let's say, challenge. So as many people, at least in the Netherlands and I think in Europe too, say, okay, my health data are owned by myself, so which is legally maybe not correct because the data on a hard disk may be somewhere else. But if I control the access, then the question is, how do I control the access? So for the data visiting, so that it can be organized. Of course, I as a patient or citizen don't want to push each time the button that yes, no, that's a weird guy, no, yes, no. So the idea is to form more a group, and you can decide which group you want to join, where you have a general agreement. I have different levels of agreement for the data visiting. Who is allowed to visit? Is it only someone treating myself, which is my doctor, and someone else who really needs the data to make me better? Is it academic researchers once they have been, let's say, passed through a different, certain kind of, let's say, good examination, whether it's really the right question, no things wrong and so on? Is it also for industry, which I'm approving? And then I can set the different marks. I can always change them. And then if, for example, a researcher asks, the researcher asks, so an agent is getting to the different points and then checking where do I have access, where not, so that privacy is always there, preserved, and the data are never downloaded, they are only visited, maybe uploaded into a very safe cloud environment, and that system would allow to share data much better. Now in the Netherlands, sometimes even the data are at this kind of central by the government funding organization put on SURF. But that's not accessible, for example, by industry, because you need an account. If you don't have an account, you can't get there. So that's what we try to change with this kind of data visiting, where there are really libraries how to access the data, so that an agent can check, okay, this request, this is someone from industry, and so on. Does he have the right approval? What does he want to do? And so on. So, and machines can communicate very good with each other because they can read each other's code or understand each other. So that's how we are doing it. And actually, that is already working in many areas in the world. Let's say in the Netherlands, we have FISMA, a company who are organizing data visiting for many, many applications, just not our health applications yet.
Thomas, one question was asked about the confidentiality of a patient information, huge. And I exemplified at the exchange, data coming from 10 centers, for example, in Alzheimer's, being deposited in massive, yes, without a complete vetting, right? There's a process at UCSD that uploads all of the data. You know, we have a whole army of people that have to be able to check the data before it gets uploaded. It needs funding, it needs time, it needs effort, so people can go through and make absolutely sure the data is completely de-identified. Nobody can connect any of this public data to a subject ID.
Yeah.
Just an issue, much complexity to think about as we move forward.
And of course, with certain data which we also want to use, let's say, suddenly you can identify who, because if you need the address and so on, and if you have a smart question, you can figure out who could be that person, or you can search even for persons because you know where they live and what they are exposed to and so on. And if you have the mass spec data, you can see what they eat, what they use. So privacy is an issue, therefore the data visiting is much more safer.
Yeah.
A lot of data is, of course, generated at core facilities, and there often the sample prep or the data analysis, data deposition are divorced or can be divorced from each other. So I wonder what could be your recommendations in that case. What could be carrots for, let's say, core facilities or labs that work with core facilities? Could there be specific grants that help us data steward? Is everything going to be from initiatives like you described? Just your comments.
Did you want to answer that? I mean, core facilities, and it probably goes back to this whole point of making sure that the data processing pipelines that are being developed both commercially or within core facilities or within the institutions work to make sure that their data is fair, accessible, readable, so that it can be just essentially a push button both for the user or the core facility to upload it to repositories in a standardized way. And I think this is what mQACC is wanting to work with, is to try and make sure that whether it's the core facilities or the data processing pipelines that everyone uses are more consistent, and that way it's simplified for everyone to upload and allows them to comply with whatever publication requirements that are emerging. I don't know, Jennifer, maybe did you want to comment on that?
Yeah, my point is, of course, that no one is going to give me that person that I need to do that work. So how can I convince my university or grant agencies that this is important to do?
So I'm going to comment on that from a core facility perspective, that I entirely agree that we should be reporting data in a way that is fair in the true sense of the word. So it has a very detailed description of how that data was collected. On top of that, we also encourage any data user to get in touch with us to actually make contact and talk about specifics of that data set. Because sometimes things come out in discussion that it's not always easy to report in a standardized form. That works when you're within a university. It doesn't work when you're dealing with 10 different data sets that you want to merge. And there we get back to standardization and numbers. The more numbers you have, the more individual samples not quite being right work, because they get filtered out.
Thank you. So please, the next question, we have only 5 minutes.
Yeah, I want to comment on the pre-analytical part in Metabolomics, because I think we focus a lot on having standardized ways of reporting analytical work, and that's very important. But I just want to remind people that, at least in a hospital, they say that more than 70% of what is going wrong with a sample happens before it enters your lab. And you have no idea what that sample has been through. So even though we have very well-defined studies, you don't know for how long was that plasma sample on the bench before it was put in the freezer. Many people involved, etc. So I think there needs to be much more focus what happens to the metabolome before it enters the mass spec or the NMR instrument. It needs to be a systematic approach, and I hope more people can contribute to do that in a very systematic way. We know that amino acids are unstable, but I mean, except from that, because yeah, so it needs to be really focused on the stability, and especially with core facilities where we have, let's say you have biobank samples from God knows when, stored in a fridge or a freezer, what temperature for how long, and also very important about controlled samples, because typically the interesting samples are stored in a freezer somewhere, and then what to compare with. So I just really want to refocus on the pre-analytical things that is hard to control and very important to characterize.
I think that's a really great point. And I think there are ways, and there are groups now producing standards and methods to compare a sample to determine how long it's been left out of the freezer, to look for certain characteristic metabolites that are known to decay. That data needs to be more public, more visible. It needs to be part of, as you say, the core facilities, but also with individual labs. And these resources need to be public so that everyone can access them. Because right now, a lot of them are sort of internal. A lot of core labs I know have their own internal sets to say, this is good, this is bad, but they don't share that. And I think this is a big problem. I don't know if there are other comments.
I think that this sample integrity, of course, is very central to any kind of standardization, and understanding that is, I think, part of these efforts as well. I think we have very little time, so please one more question and then the last question or comment.
Yeah, I just have my name is Niall O'Connor. I'm from Metaworks, where we develop an AI platform for direct interpretation of raw mass spec data. And when I'm listening to the conversations about how to use AI here, I feel like a lot of it is to try and organize data and clean up data to ultimately be human interpreted, and not necessarily machine scale interpretation. And I just want to kind of piggyback on the question that Soa asked. It's like, if you had a magic wand, what would you want a machine interpretation AI platform to do? Because we feel like it's possible to go directly from raw data to a biological insight without skipping or without going through an annotation step that's reduced by a human. And the reason that's important is there's a lot of uncharacterized metabolites in the data set. And we saw that in the lipid study that was presented. And we feel that there's a lot of that low abundance and uncharacterized data that drives the ultimate determination of biological insight.
Yeah, I mean, those, again, very good point. And I think looking at how AI will change, and that magic wand that we've talked about is, yeah, can we go from raw data to a paper, raw data to knowledge, raw data to deep interpretation and deep insight? And I think we tend to humanize it, and we want to be able to have those processes that our brains work in, but we have to have, named entities, and machine learning doesn't. And that's something that we have to wrestle with as a community, because it's not how humans typically process data. Yeah, the magic wand that I think everyone would like is something that can take the raw data and lead it to a finished paper.
Certainly a mindset change, right? To just think differently about that higher dimensional space of data that's available.
Yeah. I think Peter.
In fact, I want to follow up on the same concept in a way, and really ask the question is whether machine readable equates to AI ready. And David, you made the point about needing to link the current studies back with the literature of what's been done. And so our machine readable data now is not going to be compatible with that. And all of the large language models that are being developed, they're using language, they're not using Inchi codes or those sorts of things. And my concern is that we're going to create new languages to allow us to bring in metabolomic and lipidomic data into that framework, but we might end up with many languages. And we've just got past that in a sense, and we have some standardization that's coming through. There's a danger that we're going to devolve from that as there are multiple large language models developed that won't talk to each other. And is that being addressed, you know, at a community-wide level, I think is my question.
Yeah, I think that's a really good point. Again, we break down to this Tower of Babel with different language models, but one of the areas that probably hasn't been mentioned is that there's also things called large reasoning models, or LRMs. And these do the things that humans admire, which is to be able to take raw data or facts and systematically assemble them and describe the process of how they came to the conclusions, linking the pieces of data in that process. And you can actually see this happen if you ever try some of the ChatGPT to ask it to explain a derivation. And the large reasoning model in ChatGPT is quite good, so it's impressive to see what's possible. And I think this is where we have to go if we want to be able to sort of deal with the different languages. But reasoning is logic, and that's sort of a universal language. And if it can display the processes that it does, display the data it used, then I think we can agree with the conclusions if it's logical. Thomas, you have a point.
Maybe a small comment on that. Let's say, and maybe you can send me an email and I can send you some examples, because some of these machines, LLMs now, they also interact with you whether they correctly interpreted the data and so on. So it's not that you just put it there and you get it back, like with ChatGPT. No, no. You really have to check, for example, if the naming is not correct, do you mean this, do you mean this? Do I understand correctly that these were the settings you used? So they are very, very interactive. They just support, they're just like an assistant. And I think that's also how we should see AI. It's much more a nice assistant. And that's why in the big companies now AI is just where all the computers are at the IT help desk. So I think they can really, let's say, they should assist us, and then the different languages don't matter because they are defined, standardized, and whether you now use, for example, the European or the US standards or whatever, that doesn't matter because they are interoperable. So I think, let's say, but test them and then you will see, but maybe we can discuss about it.
Listen, I think we need to wrap up, but how do we wrap up? Please, join. Let's hear from you. Contact. You heard about so many initiatives ongoing. We are interested in human disease, in subtyping disease, finding biomarkers for disease, looking at response. If you are interested in adding the AI tools, it's a beginning of a discussion. Let us hear from you. You heard from the exchange. You heard from the Metabolomics Standards Initiative. You heard from mQACC. Lots of opportunities. Please join. I thank our discussants. I thank our speakers. Let's take a picture with the, please, the speakers and the discussants. If you can come here, we'll take a picture. We thank you all. Let's hear from you. The Metabolomics Society is your voice. Please express your vision. Put a vision for the future. It's in your hands. Thank you.